Hi, everybody. Thank you so much for joining. This is Bill Davis with Global Genes. We are going to go ahead and get started. We have a lot of great information to get through, so we want to make sure we have plenty of time. After the presentation, Dr. Amy Potter is going to be presenting the study results. After her presentation, we will be having a short question and answer session. Please use the question and answer function, and I will be reading the questions to Dr. Potter, and so just submit them, and if for some reason you submit a question after the question and answer session, we will best to get it answered, and I will send a recap of the webinar to all of the communities, and they can distribute the questions accordingly. So this is the presentation of the SD-101 Phase 3 study results to the EB community. I'm Kendall Davis, and I'm the Director of Strategic Alliances at Global Genes. I apologize, a couple of our slides, the formatting is a little bit off, but don't worry, we'll send everybody a copy of the slides as well. If you don't know, Global Genes is a global nonprofit organization, and we work to support all of the over 7,000 different rare disease communities that exist worldwide. So that we are intentional, we are connection, we are support, we are hope, and we are impact. And the purpose of Global Genes is to connect, empower, and inspire the rare disease community, the global community. Our vision of the future is a globally connected community equipped to eliminate the challenges of rare diseases. Ourselves on 3 strategic horizons. We provide versatile, individualized support for patients and disease communities. We have an free information center open to all patient advocates in their communities. And we have a model that adapts and scales and understands the importance of collaboration, connection, and sharing best practices. Everything does fits into one of these bucket areas, and we really work to provide as many resources as we can for anybody living with rare disease globally. We have a lot of fantastic resources on our website at globalgenes.org. We have a series of rare toolkits that are deep dives into specific issues surrounding living with a rare disease. We have a podcast called Rarecast. We do rare insights, which are they are complements to our toolkits, and they're just wonderful informational resources. And we also have rare webinars. So instead of doing webinars like this for specific communities, we do host general webinars for the entire rare disease community, and you can find all of that information on our website. We have wonderful events throughout the year. If you are located in the United States, or if you are able to travel, we have wonderful programs in October, in May, and throughout the summer. Again, I will send these to you because the formatting did get a little messed up and it's hard to see, but we do have a Rare Patient Advocacy Summit that happens in the fall every year. This year, it's from October 3rd through October 5th. We have Patient Advocacy Symposium, which is a deep dive into the drug development and clinical science process that happens in May in conjunction with the University of Pennsylvania's Orphan Drug Center. And we have the Road series, which is a partnership between the Every Life Foundation and Global Genes to bring awareness and educational resources to different cities around the country. We are going to turn it over to Dr. Amy Potter from Northwestern University to present the findings of the study. So the slides.
Great. I hope everybody can hear me. I want to just thank you for asking me to chat a little bit about this study that we've been working on so hard over the last few years. I'm going to start by just a little bit about EB, or Epidermolysis bullosa, but I'm pretty sure that most of you who are listening in know quite a bit about this disorder. For those who may have just joined us, I'll just remind that this is a group of rare devastating disorders that can involve the skin and the mucous membranes, and we unfortunately have no approved treatment for EB. The disorder can affect internal organs. It can even be fatal, and we know that the wounds that are part of this disorder can lead to life-threatening infections. Some are diagnosed as late as adulthood, but most are diagnosed in infancy. Overall, this is a rare disease with 30 to 40,000, possibly more, diagnosed in major global regions. We think largely as being 3 major subsets, the most common or simple the strophic type, which is with the greatest amount of scarring, and then the more unusual junctional type. Study that has been on for the last few years, and it's an exciting study because of the fact that it really was the largest double-blind randomized vehicle control study that ever has been done for me. It was a real fact that there were families who were reporting improvement with an over-the-counter cream that had low concentration of a substance called allantoin. For an open-label experience that patient-directed was 8 patients encompassing the variety of different subtypes, and it was positive, with comments ranging from improved healing to reduced pain with dressing change to fewer overall blisters. Now, this allantoin is known to be a very safe ingredient. Several studies have been done with it during the past 40 years, and it has shown to reduce inflammation, to have some antimicrobial properties, and to loosen up some of the skin bridges in skin, that is, be keratolytic. It's also shown to promote some collagen synthesis and deposition. In other words, a lot of properties that may impact wound healing. And then to an early study, the first study, and that's called Phase 2B, and the intent of this kind of a study is to try to get a sense of where it might be effective with a relatively small number of individuals participating, and this to decide which dosage to use for a larger study. So this Phase 2B, called SD003, in which 3-month double-blind, placebo-controlled, again, dose-finding study, was followed by an optional open-label extension. That is, from the beginning trial, when patients could be either randomly put on a placebo cream versus a 3% SD101 versus a 6% SD101, all then could choose, if they wanted to stay in the study, to go to the open label component. Now, the patient enrolled in this could be of any of the different types, simplex, reciproc, or junctional forms, as long as they met criteria. So they had to be at least 6 months of age, and there had to be one wound chosen that was called the target wound, and they had to be at least 21 days old and be at least a certain size, between 5 centimeters, which is about 2 inches, to 50 centimeters, which is about 20 inches. All about equal amounts, numbers of individuals enrolled, between 15 and 17, and that total number, almost all, to go forward into the open label extension, which then had 42 individuals who used it and were known to use the product. There were several assessments during the first 3-month period. Patients were seen every 3 months. The primary objective of this Phase 2B study was to make sure that this was safe. The second objective was to see if the body surface area affected by lesions was reduced in this study. And the conclusions of this study were that the investigators considered even the highest dose, the 6%, to be generally safe and well tolerated. The treatment with this for 24 months reduced the total body surface area involved in patients and down by 2.1%. And then the effectiveness and the safety data at the 6% that was seen in these studies, it was warranted to move to a much larger Phase 3 trial using the 6%, and this was called the Essence trial. So designed for the Essence trial. This 3-month double-blind treatment period with one whole body application, which is exactly what was done in the previous smaller trial. a total of 169 ED patients were enrolled. Because of safety, they were able to enroll at 1 month of age or higher. And the previous study, the now, was not just that it had to be chronic, the target had to be there for at least 21 days, but it had to be at least 10 centimeters or about 4 inches across in size. 82 enrolled who got the 6% in the double blind, 87 with placebo, so about equal numbers. And everyone optionally could extend into, again, an open label study. Endpoints at 3 months of age, at 3 months after the double blind period, working at the time to target of wound closure for a difference between the groups, and also the proportion of patients who had target wound closure. And kind of statistical analysis, it was decided up front what level of statistical significance would be, and that involved a p-value of less than or equal to 0.05. Now, secondary endpoints that we looked at, the proportion of patients with target wound closure, including time points, the body surface area of lesions and blisters, the change in the body surface area of wounds, reported itching, and patient-reported pain. As you see here, started in the United States, which had the greatest numbers with 37% of the patients, but also was designed to involve those outside the United States, a true international study. As you can see, many of the patients were also from a variety of different countries in Europe, but even Australia was involved in this study, so all around the world. Now, 110 patients originally were screened. Some did not meet criteria. Those are then called screened failures. But as mentioned, 169 patients were randomized. By completion, 155 patients left. Some discontinued, but the vast majority stayed in this study. In fact, only 3 did not enter the extension study. 152 did. So patients on the whole wanted to continue. The patient characteristics of those who were enrolled in this study. The mean groups was about equal, almost 14 years. There were about numbers of male and female, a little bit more male got the active, a little bit more female got the placebo. The wound size was about 20 centimeters squared in both groups. The wound age was around 400 to a little bit more, 500. There was a mean. 2 groups pretty much equal. And there was a variety of different EB types, but they were pretty evenly divided, with mostly recessive dystrophic individuals who were in this trial, but few numbers who had simplex or junctional subgroups. Look first at the safety results, and as you can see, the safety profile showed that what we call treatment emergent adverse events, or that were thought to be related potentially to those that developed on treatment, were generally balanced between the 2 groups. And the other ones were nasopharyngitis, that's a fancy word for cold, pruritus, fancy word for itch, and fever. So most patients in both groups got what we see most commonly, which is having an upper respiratory infection, being itchy, or having a fever. Now let's look at efficacy results. So the first endpoint was time to wound closure. Unfortunately, as you see here, you're making about the gray line for those on placebo or the blue line for those on the 6% SD101. There was no difference. If we look at other primary endpoints, the proportion of patients with wound closure, looking at the gray placebo and the blue SD101, there was no difference to 3 months and at points in the patients with wound closure. There were other results, and those also did not reach statistical significance versus placebo. We're talking about earlier points to wound closure, changing body surface area of lesions, blisters, or wounds, patient-reported itching, or patient-reported pain. There were some encouraging trends in closure observed in certain subpopulations, but those were not upfront designated as efficacy results. Many challenges in this study, which again was the very largest study ever performed with EB, and I want to applaud anybody who participated and also Amicus who put this forward, because it was really quite a feat to enroll so many individuals with this rare disease. One was that up front, the natural history of wound healing in EB was really never studied and wasn't reported in the literature, not well known. So the challenges may have been that it wasn't really clear what to choose as time points, for example. There are also very high vehicle response rates. You see this in randomized double-blind clinical trials, and it's not surprising that this occurred, because we know that the vehicle for this drug was extremely good. It itself may have had some positive effects. In the method was applied daily. Don't necessarily change their wounds daily. So increased dressing changes daily, that can have a benefit for EB. And then it's the fact that individuals who were in this study were coming in on a regular basis much more frequently than usually occurs with EB. So the careful physician monitoring all likely played a role. Another is that in order to look broadly at EB and include as many individuals as possible, the study enrolled a very heterogeneous patient population, not just in the 3 different subtypes, but even among the different subtypes. There were various severities, and there were various demographic characteristics. So, well, the study has closed out, or that is underway since September 2017, when these negative results were announced. Now a process that has been developing for submitting requests for continuing individual patient access for those many patients who enjoyed being in the study and thought it was beneficial. Extension studies, however, the SD004 and the SD008, were closed by the end of 2018. In these studies have been requested to have their end-of-study visit by 14th of September 2018 to facilitate this. As we're currently using SD101, can submit requests for continued use. There are supplies of the medication, and can continue at the open-label extension studies until the supplies are depleted. This request to be done through the physician on this expanded access program. I would encourage you to visit the patient advocacy page on Amicus's website if you want to download a copy of the policy from Amicus, expanded access of investigational drug, and please copy down this link if you are interested in doing that, and I'll give you a minute to do so. I want to make some acknowledgements. This study was the largest registration-directed study conducted to date for EB. So I want Amicus for a chance on moving forward with this topical product. And I only want to thank all those who participated in the study, the families, the many investigators and study staff, site staff, and certainly our EB patient organizations for being cheerleaders for helping all of our families with EB and trying to move forward research that may lead to new therapies for affected individuals and families. And with that, I'm just going to mention the next steps, that while Essence failed to meet its pre-specified primary and key secondary endpoints, the company behind it remains committed to sharing learning with the EB community in the hope that it will guide the development of other experimental therapies and ultimately lead to treatments for EB. And I mentioned this was really a potential learning opportunity about the natural history in patients who didn't get the active medication and certain long-term period of following these individuals. Amicus study results and key learning with payers, patient organizations, physicians, researchers and company, and regulatory authorities. So I would thank everybody involved, and I will now be very happy to open this to any questions that you may have.
So if anybody would ask a question, please use the question and answer function at the bottom right hand of your screen, and I will read the questions to Dr. Poller as they come in. So I will give everybody a minute to type your questions if you have them.
As loudly as you can. You're a little soft, even though I have my volume all the way up.
Thank you. Is that better?
Much.
Okay, just in case everybody didn't hear me, if you would like to ask a question, please type your question in the question and answer box at the bottom right hand of your screen, and I will read the questions to Dr. Poller. We'll give it just for people to ask their questions. And if you can, after the webinar has concluded, or if you go back and listen to it again later, you can email your questions directly to me, and Global Genes will do our best to get them answered for you and sent back after the webinar has concluded. Don't have any yet. Oh, we just got one. Okay. Question is, would it be possible to get the recipe for the vehicle for the cream, as this cream has had benefits for my son's skin that no other moisture ever had?
Oh, wow. Well, that's fantastic. I have to yield to the Amicus company. It is their product. And see if they have any interest in even putting it out as potentially an open, over-the-counter type of product, but I'd have to yield to them. And Kendall, I asked that perhaps you could reach out and share information later. Is that?
Yes, absolutely. We will be communicating all of the questions that are answered to Amicus as well. And if time allots after Jane Prince, we can see if she can answer that question, or if we would like to get back to the person who asked the question. Is there any other questions for Dr. Poller? We'll go ahead and take another minute or two.
One person had a question through the chat function. He asked, did I correctly understand that patients with dominant dystrophy were not included? If that's so, why?
It turned out that there was nobody in the study, but remember that there was a lower limit of the size of wounds, and they had to be there for at least 21 days. Fortunately, those who have dominant dystrophic EB, the wound tend to not be smaller, but they do tend to heal a bit more quickly than those with recessive dystrophic EB. So it just turned out that there was no one who fit those criteria enrolled in this phase 3 trial. Was certainly not discriminatory. Clearly, had it shown benefit, it was available for the entire EB community.
questions for Dr. Poller? Perfect. We'll give it just about 30 more seconds, and then we can move on. Please keep submitting your questions as we go. If we are a little bit ahead of schedule, we can come back to questions. Okay, perfect. I don't have any further questions right now for you, Dr. Poller. Thank you so much for the information and for the presentation. And now we are going to go ahead and I would like to introduce, who is the Chief Patient Advocate of Amicus Therapeutics. So Jane, I believe you have presenter privileges, so you should be able to move the slides forward.
So, good evening, everyone. I am trying to do that right now, but it seems that I don't have that ability. Kendall, maybe you should just continue.
I just did it for you.
Oh, okay, great. Thanks, Amy. Yeah, there we go. Okay. So good evening, everybody. Thank you so much, Kendall. Thank you to Global Genes for inviting me to speak to you this evening, and for everyone who's taken the time to call into this webinar and participate. We really appreciate your interest. I'm not sure that everyone has some familiarity with Amicus, so just I'll briefly give you an introduction to the company. A company that started back in 2002, although our EB program was by far one of our newer endeavors, and we entered the EB community space back in 2015. So we are a company that is really committed to improving the lives of people and their families who are affected by rare orphan metabolic diseases. And it's a commitment that we take very, very seriously. It's something that we live and breathe here at the company every day, and it is hearing and learning from people living with rare diseases, their journeys, their experiences, and how they're shared with us in patient and professional advocacy, but with the company as a whole, that really make us better professionals in development, but I say they make us better people. So for all of you who are on this call and who are connected to the rare disease community, we thank you very much for educating us. As Dr. Poller has said, the ESSENCE study was the largest EB clinical research study to date, and it was in fact the first phase 3 clinical trial. Community's commitment to move disease knowledge forward is key in any clinical research, and we certainly saw that level of activity in ESSENCE study from the different patient organizations around the world and from patients themselves, and for those where the patients are very young, from their parents and their families, their caregivers, and certainly all of the physicians dedicated to helping people with EB. And I think that this study, and Dr. Poller has said it so well, has really helped us get a better understanding for EB and what is the natural history of the disease. So this particular study did disappointing in its results, and certainly disappointing to the community, but very much so to Amicus as well. It didn't mean that we were going to pull away, but rather that we were going to lean in. And in the Amicus way, we promised that we would share the study results with the community, everybody who comprises the community. And we've done this in other clinical disease programs as well. So communicating clinical trial results is part of our commitment. The individuals, whether or their parents who participated in the study will receive patient profile results in summary from their principal investigators, so from the doctors or the study coordinators at the medical centers where they received their study visits. These results get up only after the clinical study report is completed, and we expect that to be later this year. Once the study report is done, then information from that will be utilized to help write these different individual result profiles, which will be done in lay language. So it is our attempt and our hope that this information will be easy for people not only to read, but to discuss with their physicians and to think about. We the community significantly, and we greatly appreciate all of the relationships that we've been able to build just 2015 since entering this space. And so as Dr. Paller mentioned, there are things that we're doing to continue to share with the community. So as you know, the webinar tonight, which Global Genes has organized and has taken responsibility for and done a really, really nice job, is one way for us to share this information. And I'm very glad that it was mentioned that if people have questions after the fact, if they go back in and sort of re-listen to the webinar, because it will be archived. It will be archived through Global Genes, but it also will be shared with EB patient advocacy organizations. So there are multiple ways that people can go in and listen, whether you're on the webinar this evening or whether people who couldn't make it choose to listen and learn from it. And we encourage you to let people know that it's available. You know, more questions can certainly come out. So one thing that we have been doing and are continuing to do is the development of information and resources. We do this in each of the disease communities we serve, and we certainly have been doing it in EB. So just quickly, if you look here in this bottom center, clinical trial results. So this is just sort of a template from what we've done in the past, and it's similar to what folks will see when they get their information from their PI later this year about the ESSENCE study. We developed this infographic about EB, and it opens up to a poster size, and it's available in about 20 different languages. We have since shared this material, this publication, with the organizations so that they can really take ownership of it going forward and continue to use it and post it and whatever they think is best to help get information to the community with it. We also continue to have more general, so not disease-specific information that we hope you folks will find helpful. And particularly one might be this one right here, a guide to reading a clinical or research publication. We know when folks Google for information about a particular disease, they often come across scientific literature that for the average person is not easy to understand. So the brochure is essentially a blueprint that can help people to understand what the different elements are, the different sections of a research publication that appears in a peer-reviewed journal, for example, and understand what is the best thing to take away from it. What are the key things you want to look for as you read through it? What do you want to take away from it? So for people in the EB community, as more and more research is done and more peer-reviewed journals publish information, that this may be a guide to help you better understand the information and to bring it to your physicians with questions as you become more and more educated. Our clinical trials in particular, we have found that there are some communities that know more about the drug development process than others, but at the same time, even those that have significant experience with multiple clinical trials sometimes come back and ask questions about some of the basics. So what we did was we developed this drug development process brochure, and again, takes people all the way through the clinical research process, the different phases of clinical research, and this information is available. And this clinical research certainly is informed consent, what an individual patient or their parent or guardian might give so someone may participate in clinical research. So this information for that. And this is a little bit different, and some of you may already be familiar with Our Good Stuff, and this is really an exercise in being persistently positive while living with rare disease. And it is a kit, if you will, that families or individuals can have where people are encouraged to just think about the good stuff that happens. Despite the challenges and the difficulties of living with rare disease, there are good things that do happen, and we encourage people to note those and to whether or not they use the kit and actually write these down and put them in a jar of their choice, and then periodically go to that jar and say, oh, let me go through these notes. Let me read them. Let us read them together as a family. And it can be very encouraging when you see the good things that have happened despite the challenges. And just recently, back in early February, we introduced our Good Stuff app. So you can get this from the App Store. You can search for Our Good Stuff, and it's something that you can create communities. You can speak with each other. You can have private areas. But it's just another way of thinking about the good things, being positive, seeing other people cope, how other people stay positive, and we hope that people will take advantage of that, and that certainly is available to the EB community. One of the other things that we did in the last year or so was an EB patient survey, and from this we had a poster, and I believe we have a copy of that here. So that poster, and it may be hard to see here, is Understanding Epidermolysis Bullosa from the patient's perspective. And this was presented at the European Society for Pediatric Dermatology, one of the major scientific congresses, as well as the EB Clinet conference last year. And we expect that it will be presented later this year at the European Society for Pediatric Dermatology in the spring, the poster, as well as what's called an oral or a platform presentation. And this information, it was very interesting because we asked people what was it like to do wound care, what were their needs around dressings, what was the frequency of bandage changing, what are some of the quality of life issues that make a difference for them. And so again, it's a little hard to see here, but this is information that is available for people. And we are now, next step, we're going to be developing a final manuscript from this survey, and it will be submitted to a peer-reviewed journal, we hope for publication. That journal has not yet been determined. So the other thing, and Dr. Powell mentioned this toward the end of her presentation, is how Amicus continues to share information beyond what you guys have heard this evening, beyond this survey and some of our educational resources I've covered. But we also have met individually with 5 other biotech companies who are in the EB space. Some that have ongoing clinical trials or who are developing clinical trials and soon hope to be recruiting. And work with their executives, high-level data from the ESSENCE study, and we talk with them about what we've learned, the lessons that we've learned, the good lessons, the challenging lessons, what were our best practices that we utilized to help get the study enrolled and to keep it moving forward. And we hope that they can benefit from our learnings. And as I said, looking at, similar recruitment challenges and maybe how they can avoid them. What are some best practices, things that we put into place? Well, the design of clinical trials so that we can improve the science, the understanding of EB, and move these studies forward. Additional investigational therapies. And in terms of the educational materials, we're continuing to support the community with those things, as well as supporting around other educational activities and events. There's collaboration on the public policy side of things in our work, participating with Deborah of America and their government affairs professionals and their ad hoc committee. So the rare disease priorities that are in front of our lawmakers in DC and as well as on the state level, that we can help as much as we can to make sure that EB is very visible and the needs of the community are taken into account when rare disease and other healthcare policies are considered. We have in development right now, and this came out clearly from our interaction with some healthcare providers, particularly allied healthcare professionals working with folks in EB, is a new educational resource about adolescents, about teenagers living with EB, but making their way toward adulthood and increased independence. So what are their needs? What are the things they would like to learn more about? And we're providing, so we're putting together a brochure on that. And in fact, what happened with our learning from both the healthcare providers and from people living with EB that this is a resource that they felt could be beneficial was that we are also developing a similar resource for teens living with Fabry and with Pompe, two other rare diseases that Amicus continues to work on. So with that, I'm going to thank you all. I'm happy to take questions as well. And what I would like to do is go back to the question that was asked previously, the question about is it possible to obtain or to get the recipe for the vehicle cream? As the woman who asked this said that her son's skin maybe has benefited. So this goes back to the fact that we have an expanded access program that will be available for people who've been in the study, and they can, through a physician, make a request, but we only have the medicine. We have not sort of separated out, if you will, the vehicle from the medication, which is both vehicle and active drug. So I hope that answers that question. If not, we're happy to offline afterwards get into some more detail, however we can. But if there are any other questions or comments that any of our listeners have, I would be happy to answer those that I can.
So again, if anybody has any questions, please feel free to type them in the question and answer box, and we can ask them of Jane. Somebody asked if they could have your email address. Would you be comfortable with us providing your contact information?
Yeah, what I'll do is I'll give you our, it should be on our website, but we also have patient advocacy at amicusrx.com. Get patient advocacy at amicus, AMICUSRX.com. It will come to me, but it will also come to other members of my team. So if I'm traveling or unavailable, it doesn't hold up getting your question or request in and getting back to you.
Thank you. I'm scrolling down. I see another question has come in. Actually, we had a question. Dr. Poller, are you still with us on the webinar?
I am.
Perfect. Let me just pull it up. I had a question come in right as we transitioned to Jane, and I'm going to do my best with this, but there is a word in here that I know that I'm going to pronounce incorrectly. The question is, you mentioned that NASOPHARYNGITIS.
Say that again.
I apologize. I just know I'm going to butcher this poor word. Naphrognitis. O P-A-R-Y-N-G-I-T-I-S.
Oh, nasopharyngitis.
Thank you. Okay.
So
Thank you.
naso refers to the nose, pharyngitis refers to the throat. It just means an inflammation of the nose or throat.
Sure. So the question is that you mentioned nasopharyngitis was a TE. Could you speculate on why this may be?
Well, it's so common. So there wasn't really a difference between the percentage who had nasopharyngitis or a cold, who were on placebo versus the active ingredient, versus active drug, right? But it's very, very common when you have clinical trials, and especially if those clinical trials involve children, although certainly we as adults get nasopharyngitis or colds as well. But that is really common that we see, particularly upper respiratory infections, and we saw that in both groups. So it basically said that there was nothing out of the ordinary with respect to infection or anything else that's thought to be an adverse event that came out during the trial.
Thank you. Does else have any questions for Dr. Poller or Jean Gershkowitz from Amicus? One more minute since you still have a couple of minutes. And please feel free to send your questions directly to me at kendallj@globalgenes.org if you would be more comfortable me working to get them answered for you. And we did just get one more. Question is, do you think it was the design of the study that meant the study failed to meet the endpoints, or the drug simply didn't demonstrate efficacy versus the placebo?
That's a very hard question, and you can ask that of so many different medicines out there. As I talked about, there were a lot of reasons might not have noted something when we're looking strictly, as we do in these trials, at the difference between the self, which of course may have its own good properties, the active medicine. So I think we can't really come to any kind of conclusions about whether the design, which is how you have to do it. I mean, when you have a clinical trial, you have to compare it to something. You have to have ideal vehicle itself, and that's what was done here. So for all those reasons that I mentioned, the placebo group might have done much, much better than they would have if they just weren't seeing and weren't using MACE, etc. And we just can never know that. So unfortunately, it was a negative study, but sorting out whether it was design versus the medicine is really impossible.
Thank you. Do we have other questions? Give it just 1 minute. Again, my name is Kendall Davis with Global Genes. My email is on the screen right now in blue. It's kendallj@globalgenes.org. Please feel free to reach out to me directly after this webinar or at any point moving forward if you do have questions. It looks like we don't have any other questions coming in, so I will go ahead and wrap it up. If you do have questions, please feel free to submit them the next couple of minutes. But I did just want to thank everybody. Thank you, Dr. Poller, and thank you, Jane, so very much for presenting this information to the community. We have this, the webinar as well as the slides available within 48 hours, and we will be sending them to the leaders of the EB communities that we have connected with. So however you found out about this webinar, that's probably how you will be finding out about be getting the slides as well as the information on how to listen to the webinar again. You can always, again, reach out to me directly if you would like the link to listen to the webinar, or if you would like the slides or any other information that we are here to help. So again, I don't see any other questions coming in. So with that, I will wrap it up and say again, thank you so much for everybody who was able to attend. Please look out for a recap and a link to the webinar in the next couple of days.